All right, today we are diving into one of the most powerful ideas in all of biology, recombinant DNA. This is some seriously cool stuff. It's the technology that lets us take a gene from one species, pop it into a totally different one, and basically turn tiny organisms into factories that can solve some of humanity's biggest problems.
So to really get a handle on this, let's start with the problem it was designed to solve. Because like a lot of amazing inventions, this one wasn't just a random discovery in a lab. It was born from a massive life-or-death situation. Okay, so before the 1980s, if you had diabetes, your insulin came from pigs and cows. And, it worked, kinda, but it could cause allergic reactions, and the supply was obviously limited. The challenge was huge. How do we get a pure, safe, and basically unlimited supply of human insulin? And that led to this radical idea. What if we could get something else to make it for us? Something that grows really, really fast. And that question led to a monumental breakthrough. Back in the early 70s, two scientists, Herbert Boyer and Stanley Cohen, developed a technology that let them literally mix and match genes between different species. They were, in effect, learning how to rewrite the code of life itself. Now, the formal definition can sound a bit complicated, but the core idea is actually super simple. Just think of it as a biological cut and paste. We're taking a piece of DNA we want, like the gene for human insulin, cutting it out and pasting it into the DNA of something else, like a common bacterium. So the really crucial question is, why does this even work? I mean, how can a bacterium understand instructions from a human? Well, it's all because of this profound, absolutely fundamental truth in biology. The genetic code is universal. It's the same language for you, me, a tree, and a bacterium. So when that bacterium reads the gene for human insulin, it doesn't know it's human. It just sees code, follows the instructions, and builds the protein perfectly. It's incredible. Okay, so to actually perform this kind of genetic surgery, scientists need a very specific set of tools. You can't just mix stuff in a beaker and hope for the best, right? This requires some serious precision. So let's look at the two most important tools in this whole toolkit. First off, you need a delivery vehicle, and for that, we use plasmids. These are these little circular loops of DNA that bacteria have naturally. Think of them as the perfect genetic delivery trucks. They're the vector we use to carry our new gene into the host cell. You can even think of it like a biological USB drive, ready to upload a new file of code into the computer of the cell. And second, you need a way to do the actual cutting. For that job, we have these amazing proteins called restriction enzymes, and they act like a pair of molecular scissors, but they are unbelievably precise. They don't just snip DNA randomly, they scan along the strand looking for one very specific sequence, and they only cut right there. And that, right there, is the really clever part. This whole process isn't just brute force, it's this elegant piece of biochemistry. The incredible precision of these enzymes is what makes the whole thing possible. The secret? It's all in a concept you already know. A palindrome. A word like level that reads the same forwards and backwards. Well, restriction enzymes are looking for that same kind of symmetry in the DNA code, just stretched across the two strands of the double helix. And here's exactly how that works. Take a look. If we read the top strand from left to right, we get G, A, T, T, C. Simple enough. Now here's the cool part. If you read the bottom strand from right to left, you get the exact same sequence. G, A, T, T, C. That palindromic sequence is like a big, flashing signpost for the enzyme that says, cut here. It's the perfect molecular landmark. So, we've got our delivery truck, the plasmid, and we've got our molecular scissors, the restriction enzyme. Now, we can finally put it all together and walk through the recipe for making a new recombinant organism. The whole process really boils down to 4 key steps. First, cut. You use the same restriction enzyme to snip out the human insulin gene and to cut open the plasmid. This creates these matching, chemically sticky ends on both pieces of DNA. Second, paste. Because those ends are sticky, the human gene just lots right into the plasmid. Then another enzyme called DNA ligase comes in and acts like molecular glue, sealing it all up. Third, insert. You take this new recombinant plasmid and introduce it into a host bacterium. And finally, the fun part, copy. You put that bacterium into a nice, comfy environment and let it do what it does best, multiply like crazy. And here's the magic. Every single time that bacterium divides, it doesn't just copy its own DNA, it also copies our new plasmid with the human gene inside. It's been transformed. It's now a living factory, and with every division, a whole new factory is born, each one churning out the exact protein we told it to. And because bacteria grow exponentially, one single cell can become over 1 billion cells, practically overnight. Think about that. 1 billion tiny, efficient factories, all producing pure human insulin. Problem solved.
But making insulin? That was just the proof of concept. That was just the beginning. Once the toolkit was perfected, scientists started applying it to solve problems in, well, pretty much every field you can imagine.
The applications since then have been just staggering. In medicine, we make things like human growth hormone. In agriculture, we can design crops that are naturally resistant to pests. In industry, we use engineered enzymes to help make our clothes. We can even engineer bacteria with special genes to go out and clean up oil spills and toxic waste. The possibilities are just transformative.
So the real takeaway here is this. We now have a fundamental toolkit for engineering life itself. We started this whole journey by turning microscopic bacteria into factories for life-saving medicine. And that just leaves us with the defining question for the next era of biology. Now that we can write and rewrite the code of life, what should we build next?